Welcome to Clinical Histology. I'm Doc Xie from SUMC. In these and the following sessions, we are going to discuss the basics about muscles, and we shall touch on some histological aspects of muscle pathology as well. Hopefully, by the end of today's session, you will be able to describe the organization of a skeletal muscle, the structure features within a muscle fiber, and the mechanism of muscle contraction. Before we move on, we need to know some unique terms that are often used to describe the components of muscle cells. Firstly, the muscle cells are often referred to as muscle fibers. Secondly, the prefix sarco means flesh is usually used in following terms. Sarcolemma means muscle cells membrane. Sarcoplasm refers to as cytoplasm, and the sarcoplasmic reticulum is the smooth endoplastic reticulum. Muscle tissues are composed of cells specialized for contraction, which allow locomotion, constriction, pumping, and other propulsive movements. Basically, the human muscular system is made up of three types of muscles: skeletal muscle, cardiac muscle, and smooth muscle. Skeletal muscle contains long, striated, and multinucleated cells. Their contraction is quick and usually under voluntary control. Cardiac muscle, mainly found in the heart, is a type of involuntary striated muscle. Smooth muscle is also referred to as involuntary muscle and is concerned with various spontaneous motions, such as the movement of the walls of hollow visceral, walls of blood vessels, and respiratory passages. We will have 3 sessions to talk about muscles, one for each type. We will go through skeletal muscle now and discuss the cardiac and smooth muscles in the subsequent sessions. Every skeletal muscle has 3 layers of supportive sheets of connective tissue, offering protective reinforcements to maintain the muscle's structural integrity. If we take a piece of skeletal muscle from our body, the entire muscle is surrounded by a dense irregular connective tissue called the epimysium. Inside each skeletal muscle, muscle fibers are organized into bundles called fascicles, which are wrapped by a less dense connective tissue called the perimysium. Inside each fascicle, every single muscle fiber is encased in a thin connective tissue called the endomysium. This diagram depicts a skeletal muscle fiber. The plasma membrane, called the sarcolemma, encloses bundles of myofibrils that run parallel to the long axis of the fiber. Each myofibril is surrounded by parts of the sarcoplasmic reticulum, which stores, releases, and retrieves calcium ions. The sarcolemma has deep invaginations called transverse tubules or T-tubules that encircle individual microfibrils. Each T-tubule becomes associated with 2 terminal cisternae of the sarcoplasmic reticulum to form a trias. This arrangement permits simultaneously spread of depolarization and produce uniform contraction. Although it is not obvious in this drawing, skeletal muscle actually has many peripheral nuclei. On your left is a longitudinally sectioned skeletal muscle, showing striations of alternating light and dark bands. On your right is a diagram of a myofibril. The dark bands are known as A bands, and the light bands are called I bands. The center of each I band is occupied by a thin dark line called the Z line or Z disc. In the center of each A band, we can see a pale area, that is H band. The H band is further bisected by a thin M line. Now, you can pause the video and try to identify these bands and lines on the myofibril. Each myofibril is divided by the Z-lines along its length to segments called sarcomeres. The sarcomere holds the secrets of striated muscular contraction. If we continue to zoom in on a single sarcomere using electron microscope, we will realize that the I and A banding pattern in sarcomeres is due mainly to the regular arrangement of thin and thick filaments. As shown in the micrograph, thin filaments are apparent in light or I band as parallelly arranged electron-dense threads. One of its ends is anchored at the Z-line, and the other extends towards the center of the sarcomere. The thin filaments are composed primarily of actin proteins, which harbor binding sites for the heads of myosin proteins from the thick filaments. The thick filaments occupy the dark or A band in the middle region of the sarcomere. The thick myofilaments are made up of myosin proteins, each with a head and a tail, as shown in the model figure. Hundreds of myosin proteins are arranged into each thick filaments, with tails towards the M line and heads extending towards the Z line. The myosin heads can move if fueled by ATP hydrolysis. When signaled by a motor neuron, a skeletal muscle fiber contracts as the myosin heads of the thick filaments bind to actin and pull the thin filaments. The movement of the myosin heads will drag the thin filaments towards the center of the sarcomere, which shortens the sarcomere and produces force at the same time. This process is somewhat like the tug-of-war. A muscle contracting is all about sarcomere contracting. It is the shortening of these individual sarcomeres that lead to the contraction of individual muscle fibers, and ultimately the whole muscle. For more detailed molecular mechanisms of muscle contraction, I encourage you to check your biochemical textbooks or other online resources. The dysfunction of muscle fibers may result in muscle diseases. Muscular dystrophy is a group of muscle conditions that cause progressive weakness and loss of muscle mass. Like many other diseases, muscular dystrophy can be evaluated with muscle biopsy. Muscle degeneration and regeneration are two of the most evident pathological events characterizing muscular dystrophies. The most common type of muscular dystrophies is Duchenne muscular dystrophy, DMD, which typically affects boys beginning around the age of 4 and causes death before the age of 20, usually because of lung infections and respiratory failure. DMD is an X-linked hereditary disease caused by the absence or defects of dystrophin protein. Abnormality of dystrophin protein leads to damage and degeneration of muscle cells. In response, skeletal muscle will regenerate new fibers, giving rise to many small muscle cells. Eventually, muscle tissue is replaced by adipose tissue, as shown on your right-hand side. On your left is a cross-section of a normal skeletal muscle. If you are interested in muscular dystrophy disorders, try to find more information online or from the library. Now, let's summarize what we have learned today. At the beginning, we dissected a piece of muscle. Now, we know that 3 supportive connective tissues, namely epimysin, perimysin, and endomysin, embrace whole muscle, fascicle, and muscle fibers, respectively. Then, we continued with the detailed structures of contraction unit, sarcomere. We learned how the arrangement of thin and thick filaments form the bands and the lines in the sarcomere or myofibrils. We also consider the specialized membranous structures such as the T-tubule and the triads that are important for triggering the release of calcine and spreading of depolarization. Finally, we talked about the sliding filament theory, which holds true for all three types of muscles, although definitely there are some individual differences. That's all for today. Thank you for joining us. See you next time.